3, 2, 1.
So the Calvin cycle starts with carbon fixation, one of 3 parts of the entire Calvin cycle. So RUBP adds a carbon added to it, and by the enzyme Rubisco, and Rubisco splits G3P into, actually splits RUBP into G3P, which has 3 carbons. It's a 3-carbon molecule, and it does it 3 times, making 6 G3P. And after this is done, it moves on to reduction, which is the second part of the Calvin cycle. So after this is created, you have your 6 G3P, and G3P is reduced, and it uses ATP and NADPH in order to reduce it with hydrogen to be able to create triose phosphate, and this does this to create 5 triose phosphate. Now, let's move these over here. Now we have a total of 15 carbons in these triose phosphate, and now we're in the process of regeneration, which is the last process of the Calvin cycle. So now these carbons break into 5 carbon molecules. And this is used to create more RUBP. The process of carbon fixation starts again, making G3P, and after 2 turns, there's enough carbons to make glucose. The Calvin cycle has to repeat in order to have enough carbons to make glucose, because there's 6 carbons in glucose, and the Calvin cycle has to repeat 6 times in order to be able to complete the glucose molecule, which is the whole intention of the Calvin cycle. If this does not occur, the rare occasion that this doesn't occur, the reason it would not occur is because the cyclic photophosphorylation would occur if there is a necessity for ATP and there isn't any, there's enough NADPH to go with this cycle. But if there's too much NADPH, then the photosystem 1 would be ignored, and the hydrogen would be cycled back to photosystem 2 in order to create more ATP for the cell to function properly.